Welcome to a new deep dive. This is where we take your sources, dive deep, and pull out the key insights, really giving you that shortcut to being well-informed. Today we're tackling something pretty critical in agriculture and animal health. It's about these microscopic battles, happening inside some very vulnerable animals, the neonatal pigs. We've got some comprehensive reviews here, detailed studies too, and they're all looking at this big challenge, right? These Clostridial Enteric diseases. Our mission, to unpack these really complex toxin-driven illnesses, understand their impact, which is significant, and figure out how we actually diagnose and control them.
That's right. And these, well, they aren't just minor issues. Clostridial infections, they're a really persistent threat and economically significant too. They're a major cause of, piglets dying before weaning, poor growth. It adds up to big financial losses for the swine industry globally. And it's dynamic. The main culprits, the pathogens involved, their prevalence has actually shifted over time. So we constantly have to adapt our approach. Okay, so we're talking about microscopic battles. Who exactly are the main players here? What specific agents are causing these Clostridial enteric diseases, according to our sources?
Well, at their core, we're dealing with bacteria from the Clostridium family. They're gram-positive, anaerobic, and crucially, they form spores.
Spore forming, okay. That sounds like it makes them pretty tough.
Exactly. That spore-forming ability is key. It makes them incredibly resistant, very hard to eliminate from the environment. And our sources point to three main agents causing gut disease in these newborn piglets. We've got Clostridium perfringens type C, Clostridium perfringens type A, and Clostridioides difficile, often just called C. diff.
Right, three main culprits. And what's really interesting, you mentioned this shift, is that even though they're related, how they affect the piglets, the diseases they cause, can be really different.
Absolutely. It's almost like they have different strategies, different personalities, if you will.
Yeah, like three siblings causing trouble in unique ways.
Exactly. So historically, Clostridium perfringens type C, that was the classic one, it caused this really severe, often deadly necrohemorrhagic enteritis. You'd see it strike piglets just days old. But, and this is a critical point, there's been a major epidemiological shift.
How so?
Widespread vaccination of sows against type C has been really effective. It's drastically reduced how common type C is. But that success has sort of unmasked the other two. It's brought Clostridium perfringens type A and Clostridioides difficile much more into focus. They seem more enigmatic, harder to pin down sometimes.
Okay, so let's dig into those differences. If they have these unique strategies, what actually distinguishes them? What would you see in the piglets and what's happening inside pathologically?
Yeah, we can definitely look at their unique signatures, as you put it. Let's start with type C, Clostridium perfringens type C. We could call it the rapid killer. It is. It typically hits piglets really young, like 1 to 3 days old. Clinically, the signs are severe. Acute, often hemorrhagic diarrhea, sometimes described as claret-colored. It can cause sudden death, sometimes before you even see diarrhea. And often it hits the biggest, healthiest-looking piglets in the litter first.
That's rough. And the outlook?
Very poor. Mortality is incredibly high, often approaches 100% in affected litters.
Wow.
And when you do a necropsy, the lesions are basically diagnostic, what we call pathognomonic. You see severe necrosis, hemorrhage in the small intestine, especially the jejunum and ileum. The gut itself looks dark red, purple, it's fragile, filled with bloody necrotic material and gas.
And the weapon? The toxin.
The key weapon is beta toxin, CPB. That's what drives the severe necrotizing damage.
Okay, so that's type C. What about type A? You called it enigmatic?
Right. Clostridium perfringens type A, the enigmatic commoner, it usually starts a bit later, maybe after 48 hours, can last up to a week. The diarrhea is generally milder, and crucially, it's not hemorrhagic. The feces might be creamy, pasty, yellowish, maybe some mucus, sometimes a pinkish tint, but not bloody, like type C.
So less deadly then?
Mortality is usually low, yeah. But morbidity sickness within the litter can be really high. So you get poor-doing piglets, reduced growth, more variation in weaning weights, which is economically important. Pathologically, it's much less dramatic than type C. Gross lesions are often mild, nonspecific. Maybe the intestines look a bit flaccid, thin-walled, some gas, but no hemorrhage. Microscopically, lesions can be minimal too.
And the toxin?
Its main toxin is alpha toxin, CPA. But here's the challenge, the enigma. Type A is everywhere. It's a normal resident, a commensal in the pig gut.
So just finding it doesn't mean it's causing the problem.
Exactly. Finding type A bacteria alone isn't diagnostic. That's the key insight here. Its mere presence is often a red herring. You need more evidence to link it to the actual disease. Makes diagnosis tricky.
Okay, and the third one, C. difficile.
Clostridioides difficile, the emerging colonic challenger. This one also hits young piglets, typically 1 to 7 days old. Clinically, again, mild to moderate diarrhea, usually non-hemorrhagic, can be watery, pasty, yellowish. But interestingly, you might also see significant abdominal distension, bloating, and sometimes even scrotal edema.
Scrotal edema, that's specific.
It is. And mortality, it's moderate, not as high as type C, but definitely significant. Pathologically, the hallmark lesion is quite different. It's edema, swelling of the mesocolon, the tissue supporting the colon. It looks thickened, gelatinous. The disease primarily affects the cecum and the spiral colon, what we call typholicolitis, and these often contain pasty, yellow contents.
And it's toxins.
The main ones are toxin A, PCDA, and toxin B, TCDB. Some strains, particularly hypervirulent ones like ribotype 078, that's like a genetic fingerprint, also produce a binary toxin.
Wow, okay. The rapid killer, the enigmatic commoner, the colonic challenger, very distinct profiles. That 100% mortality for type C really stands out. Let's dig deeper into the why. You said these are mainly toxemias, so it's the toxins doing the damage. What's happening at that microscopic level, and why are newborns so susceptible?
That's really the fascinating part. The bacteria themselves aren't usually invading deep into the tissues. It's the potent protein toxins they release into the gut that cause the severe damage. And the newborn piglet's gut is, well, uniquely set up for this, unfortunately. It's a critical predisposing factor.
So for each type.
Okay, for type C, its main weapon, beta toxin, is actually quite sensitive to an enzyme called trypsin, which digests proteins.
Right, digestive enzyme.
Yeah, from the pancreas. But newborn piglets have naturally low levels of trypsin activity for the first few days. And on top of that, the sow's colostrum, the first milk, is packed with trypsin inhibitors.
So it protects the antibodies in the colostrum, but also...
Exactly. It inadvertently protects the beta toxin from being broken down. So the toxin survives, targets the cells lining the blood vessels in the gut wall, elyses them, and that leads to clots, lack of blood supply, ischemia, massive hemorrhage, and that deep tissue death necrosis. It's a perfect storm, really.
A devastating vulnerability window. What about type A? Its role seemed less clear.
It is less clear. Alpha toxin can damage cell membranes, but its exact role in causing that milder diarrhea, well, it's poorly understood. It's likely multifactorial. Maybe it's just an overgrowth of this common bug in a gut that isn't fully developed yet, perhaps triggered by diet changes or maybe co-infections like rotavirus that damage the gut lining first, letting type A gain a foothold.
So more opportunistic, perhaps?
Potentially, yes. And for C. difficile, its toxins, A and B, target the colonic epithelial cells directly. They disrupt the cell's internal skeleton, the cytoskeleton. This leads to inflammation, fluid leakage, and these characteristic microscopic volcano lesions.
And why are piglets susceptible? Is it antibiotics, like in humans?
Interestingly, no. Unlike in adult humans, where antibiotics often trigger C. diff, in piglets, it seems to be mainly about their immature gut microbiota. Their gut bacteria community just isn't established yet. It lacks what we call colonization resistance, the protective effect of a diverse, healthy microbial population that prevents pathogens from taking over. Studies clearly show this. Piglets are most susceptible around 2 to 4 days old, but they rarely get sick from C. diff after about 10 days. And that timing lines up perfectly with their gut microbiota becoming more diverse and established.
So it really sounds like each pathogen is cleverly exploiting a very specific temporary weakness in the newborn gut's defenses or development.
Precisely. Type C leverages that transient low trypsin window. Type A and C. diff take advantage of the ecological vacuum, the lack of microbial competition, in the immature gut.
That really highlights something important, doesn't it? The control strategies can't just be about killing the bugs.
Exactly. They also need to support the gut's natural maturation process. That's a critical insight.
Which leads us to how these diseases spread and persist.
Yeah.
If they exploit these vulnerabilities, how does an outbreak get going? What drives the cycle? And what are the biggest risk factors that make one litter more vulnerable than another?
Right, the epidemiology. It's often a self-perpetuating cycle. The main source, the reservoir, is usually the sow herself. Even if she looks perfectly healthy, she can carry these Clostridia as part of her normal gut flora and shed them in her feces.
Contaminating the farrowing environment.
Exactly, right where the piglets are born. And then there's the spore issue we mentioned. These spores are incredibly tough, resistant to heat, drying, many common disinfectants. They can hang around for ages in dust, soil, dried manure, creating this persistent environmental contamination. So piglets get exposed right away, orally, from the environment, from the sow's skin while nursing.
And the piglets themselves contribute to the problem.
Yes, the susceptible newborn piglet acts as an amplifier. The bacteria multiply like crazy in their gut, and the diarrhea they produce sheds massive numbers of bacteria and, crucially, more spores back into the farrowing crate. This heavily contaminates the environment for the next litter that comes in. It's a vicious cycle. Those spores are like tiny armored time bombs, making biosecurity a real challenge, an invisible enemy just waiting.
Okay, so spores and sow shedding are key. What about factors that increase the risk of piglets actually getting sick?
Number one, without a doubt, is failure of passive immunity. Piglets get zero antibodies from the sow before birth. They rely entirely on that first milk, the colostrum.
Right, for those protective antibodies.
Yes. So if they don't drink enough colostrum, or if the colostrum quality is low, maybe from a gilt, a first-time mother who often has lower antibody levels, they're left wide open, highly vulnerable. Then poor hygiene and sanitation are obviously critical. If the farrowing environment has a massive load of spores, even piglets with decent immunity can be overwhelmed. And it's not just about spraying disinfectant. You have to physically remove all the manure, the organic matter first, or the disinfectant won't penetrate to kill the spores.
That's a crucial detail. Elbow grease before chemicals.
Absolutely. And finally, management and environmental stressors. Things like chilling, drafts, overcrowding, poor ventilation. Anything that stresses the piglet can compromise its immune system and general vitality, making it easier for disease to take hold.
Okay, this picture's getting clearer, but also more complex, especially with type A being common and C. diff having its own quirks. So how do vets nail down a definitive diagnosis? You mentioned the red herring possibility. How do they crack the code reliably?
That is a really crucial question, because getting it wrong means control efforts might fail or be misdirected. Moving from just suspecting Clostridial disease to confirming it, especially for type A and C. diff, really requires a systematic approach. We rely on what's often called the diagnostic triad, 3 essential pieces of the puzzle.
Okay, what are the 3 legs of this triad?
First, a compatible clinical history and characteristic gross lesions. This means seeing the typical signs in the right age group and, crucially, performing a necropsy, ideally on piglets that are acutely sick and haven't been treated yet or are very freshly dead. Seeing that intense dark red jejunum for type C or that gelatinous swollen mesocolon for C. diff, those are strong clues. They tell a story.
Got it. History and lesions. What's next?
Second, and this is really the cornerstone, laboratory detection of the pathogen and its associated toxins or toxin genes. You need to find the bug, yes, but more importantly, evidence of its weapons. So for Clostridium perfringens, labs use tests like multiplex PCR to look for the specific toxin genes. The CPB gene for type C is beta toxin, or the CT gene for type A is alpha toxin. For C. difficile, you typically test fecal samples for the actual toxin A or toxin B proteins using an ELISA test, or again, use PCR to find the genes, TCDA, TCDB, that produce them.
Okay, so history, lesions, and lab confirmation of the bug and its toxins. What's the third piece?
The third piece is the presence of microscopic lesions consistent with the pathogenic mechanism. This means histopathology examining stained tissue sections under a microscope. This is vital to confirm the specific type of damage. Is it the deep necrohemorrhagic enteritis we expect with type C, or the typhlocholitis with those unique volcano lesions characteristic of C. difficile? It helps tie everything together.
So you really need all 3 pieces to be confident. You can't just rely on one.
Yeah.
What happens if you don't use the full triad?
That's exactly right. Relying on just one piece is where mistakes happen. The most common trap is probably overinterpreting a positive culture, especially for type A, because it's so common normally. Like, you find type A in a piglet with some mild diarrhea, okay, maybe. But what if that same piglet also has severe damage to the intestinal villi, which is classic for, say, rotavirus? Then the type A is probably just an innocent bystander, an incidental finding.
Right.
The consequence of not using the triad. You could waste time and money on the wrong treatments or control measures. Maybe you focus on antibiotics for Clostridia when the real problem is viral or hygiene or colostrum intake. Misdiagnosis leads to ineffective action. Only when all 3 parts of the triad, the clinical picture, the specific toxin detection, and the characteristic microscopic damage line up, can you be truly confident in the diagnosis and choose the right path forward.
That makes perfect sense. It's a rigorous process for a complex problem.
Mm-hmm.
So given this formidable challenge, these tough spores, the vulnerable piglets, what does an effective control program actually look like? How do you protect these animals?
Yeah, it definitely requires an integrated approach. There's no single silver bullet. Effective management needs a multifaceted program. The goal is to break that cycle of transmission we talked about and boost the piglet's own immunity. First and foremost, immunoprophylaxis and passive immunity. This really hinges on vaccination, especially for Clostridium perfringens type C. It's been incredibly successful. Commercial vaccines stimulate the sow to produce high levels of neutralizing antibodies against the toxin.
Which she then passes to the piglets.
Through the colostrum, exactly. So ensuring every single piglet gets enough good quality colostrum right after birth is absolutely critical for this passive protection to work. Management of colostrum intake is key.
Okay, vaccination and colostrum. What else?
Then there are therapeutic and prophylactic interventions. In an acute outbreak of type C, giving piglets antitoxin preformed antibodies very early, like within the first couple of hours of life, can actually be life-saving. Now, treating individual piglets once they have severe peracute type C is often too late. The damage is done. But antibiotics like penicillin or amoxicillin can be effective for type A or maybe less severe type C cases if given early enough. Sometimes vets use metaphylaxis, giving a single shot of a long-acting antibiotic to all piglets at birth in high-risk herds to prevent the bacteria from multiplying in the first place. And you can sometimes use medication in the sow's feed with specific anti-clostridial drugs to try and reduce the amount she sheds into the environment before farrowing.
So direct interventions on the piglets and managing the sow. What about the environment itself, those resistant spores?
That brings us to environmental management and biosecurity. Given how tough those spores are, rigorous sanitation is absolutely non-negotiable. And remember, that means physically removing all the manure and organic debris first, meticulously.
Power washing, scrubbing.
Yes, getting it physically clean before applying a disinfectant known to be effective against spores, a sporicidal disinfectant. Just spraying disinfectant on top of manure won't work. And simple things like Hygiene, washing the sow's udder and underside before she goes into the clean farrowing crate, can also make a big difference in reducing the initial exposure for the piglets.
Makes sense. Are there newer approaches being explored too?
Definitely. Research is ongoing into emerging control strategies, things like probiotics, beneficial bacteria, to help establish a healthy gut microbiota faster and competitively exclude the pathogens. This is particularly relevant for diseases like type A and C diff, which are linked to that immature gut environment. And for type A, since good commercial vaccines aren't as widely available or always effective as for type C, some farms use autogenous vaccines.
What are those?
They're custom-made vaccines, created using the specific strains of Clostridium perfringens, type A, isolated from that particular farm, so they're tailored to the challenge on that site, can be a useful tool.
So it's really a combination of boosting immunity, targeted treatments, and tackling the environmental contamination head-on.
Exactly. An integrated, persistent effort.
Okay, so we've taken quite the deep dive here into the microscopic world of these clostridial gut diseases. We've seen the distinct threats from Clostridium perfringens type C, type A, and Clostridioides difficile. And it seems crystal clear these are complex issues driven by toxins and really tightly linked to that unique, vulnerable state of the newborn piglet's gut.
Indeed. And, the success story with type C vaccination is really significant. It shows what can be achieved. But as we've discussed, it's also really highlighted the ongoing, perhaps more complex challenges with type A and C difficile. Getting a definitive diagnosis for these requires that thorough triad approach. And control isn't simple. It needs that multi-pronged strategy, boosting immunity, hitting the pathogen load hard, and using smart, targeted interventions.
And it feels like there are still some big questions hanging out there. You mentioned the difficulty with type A finding a reliable way to tell the truly bad strains from the harmless ones, a definitive virulence marker. Plus, this whole issue with zoonotic strains of C. difficile potentially being carried by pigs, that adds another layer, right? A One Health angle.
Absolutely. The finding of strains like ribotype 078, which is a major cause of human C. diff infections, in pigs does raise concerns about pigs potentially acting as a reservoir. It underscores the need for continued surveillance and research from both an animal health and a public health perspective.
So looking ahead, where does the research need to go?
Well, definitely finding reliable virulence factors for pathogenic type A is a big goal. Developing better non-antibiotic control methods is crucial too, things like more effective probiotics, maybe prebiotics, symbiotics, and just getting a deeper understanding of that whole complex dance between the host piglet, the pathogen, and the developing gut microbiome. That's key to designing the next generation of truly effective strategies.
So the ultimate takeaway for you, our listener, it's this incredibly fine balance, isn't it? Especially in young animals, how their developing biology interacts with their environment. We've seen how tiny things, low enzyme levels, and immature gut community can open these huge windows of vulnerability. and how microscopic players, these bacteria and their toxins, can have such massive economic and welfare impacts. It really drives home a fundamental lesson. Protecting health in early life isn't just about fighting specific germs, it's often about understanding and supporting those basic biological processes, a lesson that goes way beyond just pigs. So thinking about other young populations, animal or even human, what critical windows of vulnerability might we be overlooking? Keep exploring.